Good evening, ladies and gentlemen, and welcome to the 2021 Innes Lecture. I would like to welcome Professor Dale Sanders, Director of the John Innes Centre, to the stage.
Thank you, Felicity. And good evening, everybody. It's a real pleasure to welcome you to this Innes Lecture in 2021. As many of you will know, we host this public event with generous support from the John Innes Foundation annually. This particular support enables us to invite a speaker to present a topic which is generally within the realms of history of science, and more about that in a minute or two. But just to say, for those of you who don't know, the John Innes Foundation was founded over a century ago, and since then has been supporting research and teaching in areas of plant science for societal and economic benefit. And we've had incredible support from the John Innes Foundation, for which we're hugely grateful. The Innes Lecture is just the Innes Lecture is just one small part of the support the foundation provides the John Innes Centre, and the research community both here and in Norwich and further afield is immensely grateful to them. So this year's speaker is someone whose own family history has strong ties to the John Innes Centre and in Norwich, and we're delighted to welcome Professor Nick Hopwood to this second of our virtual presentations. Nick is a historian of biological and biomedical sciences at the University of Cambridge, and is based in the Department of History and Philosophy of Science. He's been a prolific author, and his most recent book, Haeckel's Embryos, Images, Evolution and Fraud, won the Suzanne J. Levinson Prize from the History of Science Society. I just wanted to say that also Nick currently holds a Leverhulme Major Research Fellowship to write a new book, Many Births of the Test Tube Baby. So in this year's lecture, Nick will explore the links between the two challenges, those of producing images of hidden objects and those of controlling human reproduction. So this lecture, the title of which you've seen, will allow us to travel through 250 years of history to understand how we got to where we are now, and seeing images of human embryos and fetuses all around us in classrooms, clinics and on the news. So I'm really excited to invite Nick to present his lecture, and look forward to hearing what he's got to say. Before I finish, I would really encourage questions from everybody, so you can put those in the Q&A, and Felicity will field those questions after the lecture. And having spoken with Nick before the lecture, I know he'll be really happy to receive engaged questions. So over to you, Nick, and looking forward to hearing what you've got to say.
Good evening. It's very special for me to be asked to give this lecture, because I not only grew up in Norwich, I also grew up at the John Innes. My father David was head of the genetics department. That's really how I became interested in science, which took me first to developmental biology and then to history of science. So I'm especially grateful to Sarah Wilmot and the John Innes Centre for the invitation, and for taking a risk on a subject that isn't plants or microbes, at least not directly. But it is a topic that should interest everyone, because it's about where we all come from. Here's a developmental series of images representing progressively more advanced human embryos up to about 56 days. We're used to seeing images a bit like this at school, in clinics, on the news, and in pregnancy apps. People share ultrasound scans. Though debates over evolution and abortion have sometimes made the pictures controversial, most of the time we take them for granted. But go back only 250 years and human development was nowhere to be seen. How did serial images of embryos and fetuses come to stand for the course of a pregnancy? Now, I should say usage is variable, but embryo is generally used up to the end of the second month and fetus after that. Embryology aims to understand how complex forms develop from simpler beginnings. As humans, we're especially curious about our own. But human embryology is hard to study because our development is hidden inside pregnant bodies. Now, scientists have taken two main approaches to meeting that challenge. The main one has been to use surrogate species, such as chicks and domestic mammals, recently especially mice, and very recently also embryoids derived from human stem cells. But there's also a long tradition among anatomists of collecting and analyzing directly what human material they could find. The opportunities came from the involvement of medicine in human reproduction. In the 19th century, there was only dead material from miscarrying women and the occasional post-mortem. From the 1960s and 70s, in vitro fertilisation and obstetric ultrasound made it possible to view living embryos and fetuses that could develop into babies. Collecting was just the start. Technologies of visualization and communication then generated and distributed pictures that reached ever more people. Technologies including drawing, engraving, wax modeling, photography, ultrasound. To satisfy curiosity about human origins, to provide evidence for the theory of evolution, to promote birth control and maternal welfare, to monitor pregnancy, to oppose abortion, or to argue for embryo research. In other words, in making these developmental series, reproduction in the sense of procreation has intersected with reproduction in the sense of copying images. Now, making developmental series took imaging, ordering and selection, display and publication for viewing in books, exhibitions, magazines, sonograms, online video. People tend to take the series for granted as just showing how things are. In fact, they embody huge amounts of work. This slide shows a modeler finishing wax human embryos at the German Hygiene Museum in the 1920s. The series she's making, like all the others, embodies important choices. And some of these relate to the controversies you might expect about abortion and also evolution, but less politicized choices have had fundamental effects. When every image is typically of a different embryo, how is the illusion created that we see one developing right the way through? How do makers deal with gaps? Do they fill them with embryos of other animals, as is happening here, or leave them, maybe to say, please let us do more research on this? How are representatives selected, especially to use as standards? Life is a continuum, so why do the series start and finish where they do? Are they isolated from or contextualized within a pregnant body? Development often fails, especially early on, so why do almost all series show success? How has viewing been shaped by the medium? Printed drawings, wax models, on-screen sonograms, web pages. How has viewing been controlled, and what control have viewers been able to assert? Those are some questions that I hope you'll keep in mind as I sketch how this account of human origins was made. Along the way, I hope you'll find something relevant to visualizing scientific objects more generally. Plants, bacteria, and viruses are often presented in developmental series as parts of life cycles too. The advantage of human embryos is perhaps that because they've been so difficult to see and so controversial, their history makes the challenges especially clear. This is the plan of what I want to do this evening. I'm going to look at 4 broad periods of transformation when shifts in research on reproduction combined with changes in visualization and communication. I'm going to be focusing on German-speaking Europe, the United States, and the United Kingdom, where most of the research has been done. Before I launch in, I should perhaps warn you that human embryos and fetuses came into collections, as one anatomist put it, as byproducts of human distress. So I will be mentioning miscarriage and other potentially distressing complications of reproduction. So let's begin in medieval and early modern Europe, where images of pregnancy were rare, and in this dominantly Christian culture often showed the Virgin Mary. Women were distinguished from men by the periodic discharge of fluids, especially blood. If that flow stopped, then it was hard to know if it meant a healthy pregnancy or some evil stagnation. There were no hormonal tests. Most of the time, medicine wasn't involved at all. The surest sign of pregnancy was quickening, when the woman herself felt the child move inside, but it could be uncertain even then. Various schematic or symbolic images corresponded to the various meanings of pregnancy. By the 18th century, anatomical plates often included a few embryos or fetuses. Aborted eggs, as these ones were called. But they're not much arranged in series. The most famous focused on the membranes rather than the embryo, as we can see here. Things changed in the age of revolutions around 1800, when the map of knowledge was redrawn. Embryonic development gained independence as a topic, especially in the German lands. In 1799, the German anatomist Sömmerring produced the first connected series through human development, showing not just growth, but also an increase in complexity from the 3rd week to the 4th month. A countess saw the original drawings in a castle before publication. She said she had already had many children, but had never imagined their development like this. Collecting was the greatest challenge. Sömmerring's preparations came from miscarriages, spontaneous or induced. The somewhat greater medical involvement in pregnancy meant that there were more encounters between doctors and miscarrying women. But collection didn't just bring material to anatomists, it also worked an intellectual transformation. Women tended to interpret what happened as waste material to get rid of, or a child that would not now come, or sometimes as illness, or as an unremarkable late period. Sömmerring reinterpreted this in embryological terms. He then dissected to open up the membranes and reveal the embryo inside. This separated it from the connections to the pregnant body and consolidated a problematic tradition of showing isolated embryos. He drew them in a special way and selected the most beautiful drawings, as he put it, the most perfect. That was his solution to the perennial difficulty, how to construct norms from material suspected of abnormality because of its origin. Perhaps that was why it had been miscarried. Then, Zemmering said, I brought the selected representations into a certain natural order. He assembled the pictures so that it almost looks as though one embryo is developing through the series, which is ironic because the production of the series itself relied on ways in which development wasn't successful. He had his artist redraw the pictures and the plate engraved and printed in a small run, just a few hundred, for connoisseurs. Now let's think a bit more about that coy phrase, a certain natural order. Historians have debated if this is really a developmental series. I think we can see at least the beginnings of that in the microscopical drawings at the top here, that it is starting to be developmental. If so, then why then and not before? Well, Zemmering had some sympathy for the theory that there's a real increase in complexity, that everything is not preformed and just becoming visible. There was surely a general medical interest in signs independent of a woman's testimony about quickening for use in the law courts. Zemmering himself highlighted an aesthetic change. Embryos had not been seen as perfect enough to represent the human form. People had used criteria of adult beauty, but he said, we have to realize there's a criterion of beauty appropriate to each age. Do we not find a rosebud as beautiful as a rose? But Zemmering didn't just begin to show development, he also portrayed differences by sex and individual ones as well. He was a conservative, an advocate of the innateness of human inequality, who argued that the differences extended back into the womb. He assumed that all of his embryos were Caucasian, but he emphasized sex differences in the genitals once those became visible and in the rest of the body. A glance, he claimed, could distinguish the three figures on the left here as male and the other four as female. He also highlighted individuals. He wrote, does the clear brow of the 17th girl, so the last one here, not already show a certain innate friendliness? She was his favorite. He wrote of the grace of the very lovely face. The little mouth is half open. It is bordered by quite charming small lips. I'm afraid it's as though he wants to kiss them. So Zemmering took objects out of all kinds of contexts and reworked them into a series showing change as well as growth. But development is a question he raised as much as an effect that he produced. Over the following decades, through the first half of the 19th century, medics and artists copied Zemmering's images and refined the series, keeping the framework but replacing many that they came to see as abnormal. They exploited the relatively new techniques for printing drawings, but they had no human embryos from the first 2 weeks and not many from the first 2 months. So they filled the gaps with chicks and domestic mammals. As one anatomist put it, the history of the bird embryo is the ground on which we march forward. That of the mammalian fetus is the guiding star, which promises us safety on our route towards the development of man. But anatomists did study human embryos, and here the challenges were not just collecting them, but also that the preparations were tiny, translucent, and complicated. For medical students, and it's courses for them that were embryology's main home, the professors were strongly committed to visual aids, magnified and labeled drawings and models. These, from the middle of the 19th century, are on the development of the human heart. But some teachers worried that images would only confuse the less educated women who became midwives. And there were still concerns that if a pregnant woman saw these strange forms, it could adversely affect her pregnancy. So ironically, pregnant women were among the last to see them. But around mid-century, around 1850, such pictures did enter the new popular science in books and in museums. This is a handbill for one that was on show in London at Leicester Square. It does say for gentlemen only, to see this complete series of models illustrative of the science of embryology or the origin of mankind. But at some of them, women would be admitted at different times. So in this first part of the lecture, I've talked about making the first developmental series for human embryos, the start of a tradition in which there was still much reliance on surrogates and the medical courses as the main place where these were shown, but how they were also shown beyond, but with relatively limited opportunities to see them. Embryos were not part of how most people thought about pregnancy. Imagine you went to this museum. You've never seen an embryo before. How do you think you might have responded? I'd like now to talk about two major changes from around 1870 in audience and in anatomy. I'm taking them together because they didn't just happen during the same decades of the late 19th and early 20th century. They're also linked. So first, embryology was taken to the first mass audience for science. Darwinism increased its visibility because embryos provided important evidence of common descent. The most famous and notorious picture is this plate from a book intended to be generally accessible by the German Darwin, the zoologist Ernst Haeckel. The rows are for stages from fairly early to fairly late, and the columns for vertebrate species, with the last one for humans. The main point was early identity. It's also about the idea that we climb our evolutionary tree in the womb. But some said Haeckel had made the embryos too similar, especially along the top row. He was accused of forgery even, a sign of trouble that the extrapolation from other species could cause. Now, these were years in which a revolution in communication made images available much more cheaply. These plates were printed in runs of just a few thousand. Soon, you could see embryos in books with runs of 100 times that. This let embryological visions become part of a scientific view of reproduction in campaigns promoting birth control and maternal welfare. A view more oriented towards medical intervention. Now, it might seem obvious that embryos would figure, but women on trial for illegal abortion in the early 20th century often didn't talk about embryos. They said they had to tip out a waste material. Others used the Christian language of unborn children. But pictures of embryos and fetuses were widely accessible, provided they were isolated from sex. In the photograph here, visitors are looking quite interested in a maternity center association exhibit at the New York World's Fair in 1939. Millions of people saw this. But access could still be difficult to knowledge that wasn't yet taught in the schools. Novelists wrote about a student who feared she was pregnant, ashamed to have to borrow a book. a book where she could see pictures explaining what was happening. Another novelist wrote about a man deciding what to do about his girlfriend's pregnancy, running the gauntlet of a librarian determined to embarrass him. "We don't issue books of that description to the general public," she said. The second big change of the decades around 1900 is that research in human embryology was transformed. Haeckel had politicized the similarity of humans to other animals by telling the general public that all backboned animals start absolutely the same. The experts, anatomists, refused to assume similarity, especially based on Haeckel's dodgy pictures. They insisted on studying human embryos in their own right. The founder of modern human embryology, Wilhelm His, was, not coincidentally, also Haeckel's leading critic. And he reimposed order with a monumental work on the anatomy of human embryos. He still relied on abortus material collected through a network of doctors using a stick and a carrot. The stick was that if gynaecologists kept embryos and published their own analyses, then he would criticize these as incompetent. The carrot was that if they handed over their treasures, as he called them, he named these after the donors. The gynaecologists, that is, not the women from whose bodies they came. By competent, His meant thorough internal as well as external descriptions. The precious objects had to be sacrificed, as he put it, by slicing them up completely on a microscopical guillotine that he'd invented, otherwise known as a microtome, and then rebuilt as magnified models. So this montage shows the steps from photography through drawing the whole embryo, serial sectioning, perhaps a graphical reconstruction, and then freehand modeling with a lot of measurement of the embryo in wax, and then the commercial production of these series, also showing the anatomy inside, that were sold and bought pretty much wherever embryology was taught. His constructed normal development from material which was perhaps not so likely to be normal by relentless comparison and criticism. And this normal plate embodies his reform, showing embryos from the end of the second week to the end of the second month. So he criticized a great many preparations, including many old favorites. He even rejected one embryo after anatomists had debated for 7 years whether it was normal or abnormal, as not human at all, but a bird. That shows how open things had been. But now, with Hiss's framework, anatomists competed to find early specimens, cooperating with gynecologists to fill the gaps that the series defined. They prided themselves on doing human embryology on human embryos. In 1914, American students of Wilhelm His institutionalized this human embryology in an institute funded by the Carnegie Institution of Washington, the first research institute anywhere devoted to embryology. They expanded the approach as reproduction was moving into the hospitals. So they were supplied with higher quality material from gynecological operations through a bigger network of physicians. His had collected dozens. They amassed 9,000 in a fireproof vault in Baltimore. That's what we're seeing here. Microscope slides on the left and on the right, cabinets containing the written records. They also aimed to make a difference in clinics that were increasingly a place for antenatal care. They promised to shed light on pathologies of pregnancy and on infertility. They produced a large body of detailed work, including a lot of models. So here's a plaster model of one of the Carnegie embryos, which we're seeing in 4 views from 4 sides. See how detailed it is. It's built up from slices, highly magnified. It's a stage 10 embryo. They also set up the standard staging system that's still in use, which was very tricky because of the amount of normal variation. Now, there would be quite a bit more to say about their work, including the use of monkeys as a yardstick for human development and a project in so-called racial embryology. But I'll just mention how they retrieved specimens through the first 2 weeks. Between the 1930s and the 1950s, researchers at Harvard Medical School collaborated with the Carnegie Department. This so-called Boston egg hunt arranged clinically indicated hysterectomies, operations to remove the uterus, at known times after menstruation and intercourse. Now, that was ethically delicate then, and it would, of course, not be ethical today, but it increased the chance of finding something. This is a photograph and drawing of the Harvard egg. At 12 days, it's just recently been implanted in the wall of the uterus. By around 1950, these researchers had closed the gap at the beginning of human development and had elaborate descriptions almost right through. Reviewing the material from the egg hunt, they found that over 1/3 of the embryos were so abnormal that they wouldn't have developed much further. This textbook figure is a bit more complex than the usual series. The Carnegie researchers had also, in the work on monkeys, confirmed the menstrual cycle. This figure places the ovarian cycle and an early embryonic series on a diagram of the female reproductive system, and so brings just a little of the woman's body into the picture. It indicates how cycle and series are linked, with an embryonic series only if fertilization happens. It doesn't let on that there's still a rather high rate of wastage, even if fertilization does occur. Okay, so in this second part of the lecture, I've talked about how developmental series of human embryos were controversially taken to large audiences, and about how in response research on human embryos was set up as a distinct field of anatomy. Now, just as that research had reacted to public debates, so it also fed into them. After World War II, photojournalism took Carnegie embryos and fetuses into glossy magazines, and some appeared most famously on the front cover of Life magazine. That was in 1965. They were introduced with human reproduction into school curricula around the same time. This spread is from an American textbook with a prize-winning photo from the Carnegie Department. In the UK, human embryos came in through Nuffield Biology. From around 1970, these photos began to be exploited, especially in the United States, in campaigns against the reform of the 19th century abortion laws. Anti-abortionists read development backwards to make fetuses and even embryos into babies. Legalizing abortion gave researchers more material, but provoked a backlash that has mired prenatal development in controversy ever since. Now, the Carnegie researchers had won the argument for studying human embryos directly, and fresh supplies of material and new modeling techniques gave them plenty to do for a time. But after World War II, anatomy went out of fashion. Instead, developmental biologists learned a lot from experimental, cellular, and molecular studies that were impossible on humans. They used model organisms, especially mice. Mice had been engineered for genetic research, and by the 1960s, their early embryos could be cultured. But there were new views of human embryos in this period, and they came from research in hospitals and out of projects to monitor and control reproduction more tightly. I've been talking mostly about anatomy. I'd now like to talk about live images. There were two key developments: ultrasound monitoring and in vitro fertilization. The result was that obstetricians and embryologists were selecting not drawings or photos that looked back to dead specimens, but images of embryos that could develop into babies. Let me say something about ultrasound first. The movement of childbirth into hospital allowed increased antenatal surveillance, and obstetricians discovered a fetal patient. Fetuses had been imaged by X-rays before, and various other techniques were being tried, but it's ultrasound that took off, and obstetrics today is hard to imagine without it. Malcolm Nicholson and John Fleming's history of Ian Donald's program in Glasgow shows how sonar, developed for submarines, was applied to pregnancy, beginning in 1958. The slide shows an early ultrasound image. For a long time, it was impossible for lay people to interpret such things without a great deal of help. But gradually, outputs became more pictorial. They could be put on television monitors and seen in real time. As scans became routine during the 1980s, they accelerated the experience of pregnancy and reduced the importance of birth as a threshold. Now, the Glasgow team calibrated their results against anatomy, the anatomy that I was talking about before, and argued that their own numbers were better. But coping with normal variation and distinguishing it from actual problems is a challenge still, and the stakes are quite a bit higher when working on living patients. The central contradiction of ultrasound consultations is that a sonogram usually makes a pregnancy more real. It's been called a technological quickening. But the main medical purpose of the scans is to screen for the problems, malformations, growth restriction, that crop up in a few % of cases. This is an example. Nuchal transparency, or the size of the fluid-filled space at the back of the fetal neck as a risk marker for Down syndrome. To make this kind of diagnosis, consultations draw on the population level data that informs the measurement algorithms loaded into the ultrasound machines. These standards are debated. Should they be universal or tailored to different ethnic groups? Which approach will better uncover the effects of poverty and so help reduce globally high levels of neonatal mortality? Ultrasound is also a key technology of sex selection, and mainly not, as Zermering might have used it, to select beautiful girls. Now, through the same period, human embryos began to be cultured in vitro. This started in the 1940s as part of the Harvard egg hunt and culminated in 1978 with the birth of Louise Brown, the first so-called test tube baby, and the making of the reproductive biomedicine industry. What that meant, the change that this meant, is best illustrated by a comment from the Cambridge physiologist Robert Edwards at the press conference in Manchester the morning after Louise Brown was born. "The last time I saw her," Edwards said, "she was just 8 cells in a test tube. She was beautiful then, and she's still beautiful now." IVF had introduced the possibility of a prospective view of seeing a particular egg and then early embryo that would go on to become a baby. Except that most of the time it didn't. didn't always in nature, as embryologists knew, not least from the Boston egg hunt, but rarely represented, and it certainly didn't always in culture. To improve the success rates of IVF, embryologists selected embryos for replacement into the uterus. What the Carnegie department had done with dead preparations and drawings, they did with living embryos. Since the late 1980s, it's become standard to give embryos grades for appearance, almost a criterion of beauty like Zimmermann. This 8 cell is especially clean and symmetrical. It's a grade 1. But actually, these grades were not a great predictor of successful implantation. Implantation in the wall of the uterus after the embryo had been transferred. Many clinics are hoping that time-lapse microscopy will produce better results. So this is remote imaging within an incubator that enables real-time monitoring of development and scoring against dynamic indicators of quality. I think it's fair to say that the jury is still out on how much this is going to help. So in this third part of the lecture, I've talked about the remarkable ways in which ultrasound and in vitro culture made it possible to see living human fetuses and embryos that would usually or sometimes develop into babies. This brought images of embryonic and fetal development into the heart of making families. And for that reason, I expect that many of you will remember, perhaps with some emotion, encounters with pictures of this kind. But what about human embryology? Although publicly controversial, research on human embryos was still rather marginal through the 1970s and 1980s, when model organisms made most of the running. But human embryology has gone through a revival since then, and this is producing prominent new images of human development. I'd like to finish by saying just a little about this change. After much debate, it was agreed in the UK that spare embryos could be used for research up to a limit of 14 days under strict regulation enshrined in the Human Fertilisation and Embryology Acts 1990 and 2008. As a result, a lot more has been learned about development before implantation, but the revival of human embryology was broader than this. As the internet was starting, projects were launched to digitize old slide collections, especially the Carnegie collection. That's where these are from. The virtual human embryo series made by digitizing the old Carnegie slides is on websites and in apps. But it shows only anatomy. To study gene expression needed fresh material. Geneticists wanted to go from the structure of genes to discovering where they were active, which they hoped would show how development works and how it goes wrong. So they're renewing the argument that to understand what makes us human, they had to study human embryos. And by 2000, they had the human genome sequence, which made research on human embryos easier. In 1999, they set up the Human Developmental Biology Resource. So it's based in Newcastle and London to supply embryos and fetuses in the old way from terminations of pregnancy, but now within a modern ethical framework and with various other factors making the embryos more likely to be normal. Researchers use specific molecular stains to light up regions and cell types and new kinds of microscopy to detect them. The laboratory of Alain Chedotal in Paris has been producing especially striking images. These ones are of the peripheral nervous system, shown using light sheet microscopy. So these are virtual 3D models using not sheets of wax, but optical sections. starting around the same time, the end of the 20th century, human embryonic stem cell lines were derived from spare IVF embryos. Then it was shown that stem cells could also be derived from ordinary adult cells. And recently, embryologists have learned to make them differentiate, not just into disorganized cells, but also into so-called embryoids. These stand in for embryos at stages later than in vitro culture is allowed. It's all about seeing in setups designed for fluorescence microscopy and reported online to specialists and to newspaper readers. In a way, embryoids take us back to surrogates, only now the surrogates aren't just other species, particularly mouse. They're also in vitro systems that use human cells to model aspects of human development. There are still questions about how relevant those models are. There's a lot of excitement about all this, but I hope it's clear that it builds on a long history of seeing human development. And I've tried to give a sense of that history tonight. These images were produced and viewed at the intersection of two sets of changes, in reproduction in the sense of procreation, and in reproduction in the sense of copying images. Medical engagements with pregnancy and miscarriage, contraception and conception shaped how the material was collected and how the images were used. Technologies of visualization and communication generated pictures and multiplied opportunities for viewing. The results were developmental series that, I hope I've managed to show, embody constraints and choices that shape how human development looks, whether gaps are hidden or filled with other animals, or researchers say, we've just filled one, give us some more money, maybe change the law, and we'll fill another. How representatives are selected as normal and as standards, how isolated or contextualized the series are, how the series almost always show successful development, how much the eye is encouraged to linger on one image or to track quickly along a series, where the series start and end if they're read forwards or backwards, and much more. Thanks very much for listening. I look forward to your questions.
So welcome to the virtual John Innes Centre. I'll try and join you on the screen in a second, but you are now live to our audience. I had a few questions in, so unless there's any comments you wanted to make, I'll start with the questions.
I think people have heard a lot from me already, so I'm quite happy to go into the questions. Okay.
So one of the first questions we had in related to some of the earlier work from the German lands, as you called them. They were curious to understand and know more about how the developmental modelling traditions from the German lands influenced American embryologists and how those techniques differed over that transition.
Okay, well, that's a lovely specific question, and I mean the answer is that the American researchers responded very directly to the techniques that they often originally learned by going to Germany. But then at the Carnegie department, they developed new versions of those, so that, for example, instead of stacking up wax plates and cutting away the material they didn't want to reveal the model, they did the opposite. They developed a lost wax process where they'd stack up the wax but cut out the embryo from the middle of each plate and then pour the plaster into that mold. And so that plaster model that I showed is made in much greater detail than was ever achieved in 19th century Germany by that method.
Interesting. And did that lead on to then the technology development that happened to bring the sort of the images that we sort of know now, or how did that transition?
Well, conceptually, the way that we're now used to, I mean, in embryology, but also in lots of other fields, building up virtual 3D models from optical slices. I mean, what we're looking at with these wax modeling techniques, they're the ancestors of that way of doing things.
Great, thank you, Nick. So we've had a couple more questions in while that's been happening. So one question from an anonymous writer says, do you know if medical students were taught to think critically about the images or that they were presented with in lectures?
Well, I think it all depended on the teacher, perhaps on also how the students responded. Teachers were worried about certain difficulties in trying to communicate these complicated structures. As they got more complicated, they changed in shape. And so one thing they worried about was that when students would see the image of a section of a slice in a textbook, and then they would see another slice, they would somehow think that what happened was that one slice turned into a slice at an older age. So there was effort to think critically in that sense. Was there much teaching of, if you like, a more critical approach, also taking account of some of the more political issues I've been talking about, where the material came from, the choices that were made in constructing developmental series? Well, I think some of those were thematized, but really only very occasionally. A lot of that more critical approach really only comes with the revival of feminism and new scholarly approaches from the 1970s.
Great, thanks, Nick. Very thorough answer. It's quite a long one, so I'll read it all out. It's got some comments as well. So thank you for your fascinating lecture. Throughout, we have shown many images rendered in different media. The Anatomical Museum Basel montage is a good example here. Can I ask you to reflect on the relevance of the media used in any given image? Are there differences in the kinds of medico-scientific knowledge produced depending on the media used? Does the media of an image affect the meaning of the subject it purports to depict?
Okay, so that's a very interesting question. I mean, certainly one can see how say the use of the wax modeling from serial sectioning made it possible to see inside the embryos in a great deal more detail than had been possible just by dissection. And I could talk more about ways in which anatomists, obstetricians, others learn different things from different media. But it's also interesting to think about it from the point of view of the viewers. So it makes quite a difference if it's a medium that you can view on your own, for example. So in that novel where actually it's a chemistry student who fears she's pregnant, goes through the embarrassment of borrowing a secondhand book, and she can take it down to the river and have a look at the pictures on her own. That's a very different sort of scenario from the kind of group viewing that was happening, in a room full of other people at that exhibition in New York. So media have an enormous effect in the way that different relations are set up between people looking and people showing, and I think that's one of the most interesting aspects.
Yeah, I think one of the other questions actually links into that nicely, around when the first images or reproductions of embryos were shown in the 1850s. What was the public response, and how did people respond to the displays of human embryos? Were they revulsed, or was there disbelief? Did people believe that these were real embryos?
it was very variable, and there are certainly examples of disgust, and quite often we find reports in the historical record of people disgusted, but there's also a strong emphasis, and I think this comes out nicely in the way that Zermering stresses the beauty of the embryos, that these are beautiful, and they're beautiful in their own way. They're not beautiful like children or like adults, but there are criteria of beauty for the different ages, and that's something that embryologists have done a lot to stress, and also in presenting some of the images. Photographers and others have put work into aestheticizing, into adding color, into trying to make them look less anatomical, and so on. So that's absolutely something that you see, and so there are responses to some of the magazine publications in the 1960s, where readers write in and say how beautiful, how what a privilege it was to see this, and certainly that's something that we're still familiar with, I think.
Were those sort of standards of beauty different from the kind of social construct of beauty at the time, or were they kind of linked to how beauty was seen then? Or is it something totally different? I
mean, that I find a difficult question, because does it, in a way, I guess what I'm saying is, if there were criteria of sort of what counted as, say, fashionable beauty, what shape people, adults, maybe especially women, were supposed to be, what the anatomists are saying in a way is forget all that. But it would be interesting to see if maybe in certain periods embryos are supposed to be, say, chubbier or arranged in different ways, and just whether there is some correlation with standards of beauty in another cultural field.
Yeah, that would be interesting, I think. I'm not sure how that would go, but I've noticed that Dale has unmuted his microphone. I was wondering if he wanted to ask a question or if he was just listening.
I did. Thank you, Felicity, and thanks, Nick, for a wonderful seminar. I had two things to say. First of all, thank you again. That was really stimulating. It's not. As you know, it's not JIC kind of mainstream, but everybody wants to be stimulated by biology, and that's the case. But I just had a broader question, without trying to usurp other people who want to come in. It's what about the history and the past do we want to take forward to the future? What do we want if you look ahead, say 2 decades with embryology? Where do we want to go with this in the new era of, of course, justifiable feminism and all of that? Where do we want them? Where do you think that should be played out? And it's a sort of an ethical question, not a biological one.
I mean, as you probably know, there's a great deal of discussion around that at the moment, because as embryologists are learning to culture human embryos for longer periods, there's pressure to start considering a reform of the 14-day rule. And so this is being, and I'm sure will in future be even more actively debated. I think the contribution that a historian can make is, in a way, less to say what will or perhaps even should happen in the future, but to say, look at the past as a resource. look at the way that what is often presented as completely new, it actually has quite important precedents, and not only in terms of how much knowledge had been produced, but also some of the ethical issues. Not that they should be done in the same way, but that thinking about some of the issues and thinking about what now strikes us as unacceptable, how to avoid being in that sort of position ourselves. So I think I'm offering history as a resource for a longer view, an opportunity to stand back and see this in a larger context. Of course, it'll matter to talk about particular pieces of legislation and what should be allowed and so on, but to get a sense of the enormous change and some of the ways in which that's happened. And I think also more specifically, what I've been suggesting in the talk is thinking about, I've highlighted developmental series, the way in which quite important positions are taken by the way that those are set up, and that sometimes the important things are happening not where most of the discussion is, but somewhere else. So important to look at the whole process, at what's perhaps not in the limelight, and that's I think where that longer view, that sort of standing back from the kind of cut and thrust of the immediate debate can maybe be useful.
Thanks, Nick and Dale, for those great questions and answers. We've got quite a few more questions coming, so I will crack on with some more so we can cover as many as we can. We might not be able to get to them all today, but we will send them all to Nick, and we'll try and answer them on social media and other places. So one question that came in, you mentioned that the Carnegie Institute also had a project that focused on race and genetics. I'm wondering if you could say more about the link between 20th century embryology and the ideas around eugenics or race.
Yes, so I did mention that the Carnegie department did some what they called racial embryology. Now, this wasn't a particularly large area of the work there, and in general, issues of eugenics, race, and so on have been much more connected to the history of genetics in the 20th century than to the history of embryology. But they were interested, the anatomists there, in differences between Caucasian, African American, Native American. I mean, these aren't the terms that they used, and also they had quite a few Filipino embryos, and so they did measurements. They tried to look for differences. They didn't come up with a great deal by way of conclusions. One of the main pieces of work by an anatomist called Adolf Schultz used these results really to argue against theories that different human races evolved separately. He argued that actually these results confirmed a common evolutionary origin and supported the theory of evolution. So that was just a little bit about that racial embryology project.
Great, thank you. Okay, so next question. Do you think Ernest Haeckel's illustrations are scientifically significant and influential, or should they just be seen purely as art?
Well, they were certainly highly influential, and were, for many people, important evidence of evolution and also aesthetically attractive at the same time. So for Haeckel, science and art really went very, very strongly together. Of course, if you ask, were they accurate? Should we still use them today? Then no. I mean, they have quite serious inaccuracies in them, and since there are much more accurate illustrations available, we should be using those, unless you're giving a history talk, say, where I think some distancing is understood.
Yes, I think so. Very good observation. Right, we've got loads of questions, so I'll try and pick some ones that are a bit different. Thanks for the lovely talk, Nick. How much attention has been given to the extra embryonic structures? You mentioned membranes in some of the earlier images. What about the placenta, umbilical cord, etc.? They don't seem to feature in many of the images.
Yeah, that's a very interesting question. I mean, certainly I did focus on the embryo and fetus proper, as it were, and that is, I think it's fair to say, where most of the scientific effort went. But the membranes have been, and questions about the placenta have been of great interest to some groups of researchers really throughout this period, and I mean more at some times than others. So I mean, in the 18th century there was still a lot of interest in the question of, were there nervous connections through the placenta? Could that be one route for the effect of the maternal imagination on the fetus? Lots of debate still in the 1880s, and something that Wilhelm His is much involved in, around the question of the allantois. Did human embryos have a free allantois? That was greatly debated. So and so on. So that's absolutely there. It is part of textbooks of human embryology. I've said less about it tonight, in many ways for reasons of simplification. Also, I think because those medical students, and there unfortunately have been some who don't find embryology thrilling, it's often the membranes that they find it hardest to get their head around.
Interesting. Another slightly different question. You said you wanted some difficult questions. So can you tell us a bit more about how feminism has engaged with the visualization of embryos? Has it given women more or less agency in their pregnancies?
So certainly feminist activists and scholars have engaged greatly with the kind of images that I've been showing, though much more in the context of debates over abortion. So in particular, Rosalind Paczewski's article from 1987 about the power of visual culture and the politics of reproduction, that launched the idea of the public fetus as a new actor through the visual displays that were going on, and particularly this is in the time of Ronald Reagan's second presidency, that were equating embryos and fetuses to babies, and so there have been a lot of eloquent critiques of that, and an increasing move to look historically as well, and to produce some of the work that I've actually been building on in preparing this talk and in the book that I'm writing on this topic.
Great, thank you. So a couple more questions if you've got time to answer them. So
absolutely, no,
time for a couple more. Someone said, I know you said you'd focus on European and American histories, but I wonder if you could comment on Eastern or other cultures' understanding and visualization of human development during a similar time frame. I'm guessing that could be a whole another 40-minute talk, but if you've got any key points that you could give us now, that would be great.
Yeah, well, maybe just make 2 points. The first is that this view of developing embryos that I presented as coming out of initially late 18th and early 19th century Europe, and then being elaborated. I think that is something rather distinct from the understandings of coming into being of humans that existed in Europe before then, and that were common in other cultures, in other parts of the world. So I do think this is distinctive, but certainly by the time you get to the 20th century, and especially the period after the Second World War, then this is becoming a much more international field, and actually the largest collection of human embryos and fetuses in the world is in Japan, made possible by the unusually liberal abortion laws that were established in Japan, and that made it possible to. And what's important about this collection is that they were not just from women who were seeking medical help, but this was the whole range of abortions. So it was very useful in trying to estimate rates of abnormality and so on.
Great, thank you. I think I answered one of our other questions around the influence of the Kyoto collection, which was asked as well. So 2 questions.
That's right, that is the one, yes.
Yeah, great. So we've got a couple more here. Do you know of how these images were popularized and received by the general public, especially mothers or mothers-to-be? This period follows the heels of scientific motherhood in America. And what were the religious and or moral dimensions and responses to these images during that time?
So it is very much in the context of campaigns to educate mothers and mothers-to-be that these images are being shown. So once antenatal classes start, then these sort of series were shown, and it's all about encouraging people, and especially mothers, to think in more scientific terms about what's happening. So lots of examples of attempts to communicate. Much less is known about what the audience is made of that. And so I certainly wouldn't want to be understood as saying that everyone just accepted what they were shown or told. So there's lots of evidence. There's certainly enough evidence that how people think about what's happening during human development can be really quite different from the way that embryologists think about it, even today.
And were there any kind of religious and moral quandaries that people had as a result of them? Well,
one of the important ways that religion plays in is in debates over abortion, depending on the particular Christian denomination or other religion, depending on when that was. But one of the points about the making of this view of human development is that, in some ways, what's important about it, I mean, what makes it and what gives it its authoritative science, it's you pretty much, after a certain point, you pretty much have to accept this. There are still debates to be had and arguments about what things mean. There are ways of presenting images differently that highlight one aspect or another, that draw equivalences with babies, say, or otherwise with other animals and so on. But there's only so much latitude, really, thanks to the work that has been done to establish this. You really, whatever your religious beliefs might be, at some level, you have to accept quite a bit of it.
Great, thank you. So we've got a final question. I think we've got time for one more. And he says he's usually keen to discourage my students from slippery slope arguments because they don't have any logical basis. However, do you think this is an area where attitudes have become less ethically rigorous as technology has improved?
Interesting. I mean, I'm not an ethicist, but thinking about that historically, I mean, I think one could make the opposite argument, that as more has become possible, and that has gone hand in hand with more public communication, that, I mean, ethical guidelines have certainly got stricter and more detailed. And the regime in the United Kingdom, with a rather permissive but highly regulated setup, is often held up as an example to other countries. So I would actually see I would see the guidelines becoming tighter as more has become possible. But as I was saying to Del Sanders before, I mean, maybe more focus on the specifics and the details, and useful sometimes to stand back and take a longer view.
Thank you. Yeah, that's really interesting. And thank everyone in the audience for all of the amazing questions that have come in. There are a few more that we haven't managed to get to, but I think we're going to call it a night now, because we've had an excellent discussion, and I'm sure people will want to get on and sort themselves out for the rest of the evening. So I'd like to thank you again, Nick, for coming and speaking as this year's Innes lecturer. It's been a real pleasure to hear about your research and to see more about the visible embryos. So I think with that we'll call it a night. So thank you again. Normally we'd do a round of applause, but
thank you very much.
Yeah.
Thank you for having me, and thank you everyone for the questions. And if I get a chance, I'll be happy to do my best to answer any that we haven't got to yet.
Yes, I'll send them over to you after this. Thanks.